Hello everyone, welcome again. This video is a tutorial on how to search and retrieve 3D structures from PDB. And as a norm, before we dive in, let's understand the concepts. PDB, the Protein Data Bank, is one of the oldest open access biological databases in the world. It was established in 1971 at Brookhaven National Laboratory, and it currently holds over 230,000 biological macromolecule structures, with every entry having a 4-character PDID. And just like the NCBI have ascension numbers, in this case we have PDB ID. For instance, 1 TUP indicates P53 protein bound to DNA, and 4 HHB indicates the human hemoglobin. So why do we need these 3D structures? DNA sequence tells us about what a protein is made of, while the 3D structure tells us how it works. It is as simple as that. The shape determines the function. You can see it as the protein 3D folding telling us what it can bind to, what it catalyzes, and how drugs can target it. Another thing worthy of note is how these structures are determined. The first one is by X-ray crystallography. This is the most common method. Protein is taken, it is coerced into forming a crystal, and X-rays are shot through it. How this X-ray diffract is then measured, and from the diffraction pattern, the 3D arrangement of atoms are reconstructed. About 85% of PDB structures come from X-ray, and the quality is described by a number that we call resolution, which is measured in angstrom. 1 angstrom is equivalent to 1 to 10 billionth of a meter, and anything below 2 angstrom is known to be excellent. At that resolution, we get to see individual atoms clearly. Anything above 3.5 angstrom gets blurry. These are numbers you should check before you trust a structure. The second way to determine these structures is by nuclear magnetic resonance spectroscopy. With NMR spectroscopy, instead of crystals, proteins are dissolved in solution, and how the atom responds to magnetic fields is measured. It works beautifully for small proteins, and it is particularly good for capturing the flexibility and movement of things we can't see in a crystal. The third method is by cryo-electron microscopy, cryo-EM. In this method, we flash freeze the protein sample by firing electrons through it. Thousands of the 2D images are collected from different angles and computationally reconstructed into 3D structures. This method is the reason we now see the structure of the entire ribosome, viruses, membrane proteins that were impossible to crystallize. The fourth method is AlphaFold. AlphaFold is an artificial intelligence system which was developed by Google DeepMind and EMBL-EBI. It predicts a protein 3D structure from its amino acid sequence. It is one of the most significant breakthroughs in modern-day structural biology. Now that we have established this, let's dive deep into the practical aspect. It is hands-on, and I will employ us that we practice along as we navigate through the sequence of retrieving 3D structures. The first thing you do is to open your browser. Go to rcsb.org. And welcome to the homepage of the Protein Data Bank. While we wait for it to fully load, you can explore what the page looks like. We have deposit, search, visualize, analyze, download, and learn. It is similar to the workflow on NCBI page to understand the concept. Over here, you enter your search bar and we'll be searching for the human hemoglobin. It carries oxygen in our blood, and it is one of the most iconic structures in structural biology. So in the search bar, type 4HHB, 4HHB, could be capital, 4HHB, and enter. Let's study this page. The result is up, like we did for sequence retrieval. Let's study the page. The title tells us that this is human deoxyhemoglobin, hemoglobin without oxygen bound. The crystal structure of human deoxyhemoglobin at 1.74 angstrom resolution. That already tells us that it is deoxyhemoglobin, hemoglobin without oxygen bound. The method is X-ray diffraction. The resolution is 1.74 angstrom. This is excellent, which means that the structure is high detail. Now, let's also explore some sections on the page. We should look out for the macromolecule section. Hemoglobin is a tetramer. It has 4 protein chains working as a unit. We get to see the 4 chains here. The first chain is A and C. A and C chain are alpha subunit alpha, while the other 2, B and D, are the beta subunit. Note that a single PD entry can contain 1 chain or dozens. This is relative to what type of atom it is. Is it tetramer? Is it dimer? And all of that. The other section to look out for is the ligand section. The ligand section, we can see HEM. This is the hemi group, the non-protein part of hemoglobin. And you get to see a ring-shaped organic molecule with iron. If you observe closely, there's iron at the center of this organic molecule. The iron is what actually binds to oxygen. And in this particular protein structure, we have 4 hemi group, 1 per chain. E for A, G for B, H for chain C, J for chain D. And in the PDB file, non-protein molecule like these are marked with HET ATM records, which we get to see when we explore the structure. Now, you can scroll back up to explore. And over here, you can click on the 3D report. Wait for the report to come up. We're waiting for the report to pop up. A 3D report is up, and the ribbon structure you are seeing here represents the protein backbone. We can explore this, zoom in on it, carefully over it, zoom in. You can use your hand over it, click it, turn it around. The coiled section are the alpha helices. This one is coiled. Anyone that is flattened out is the beta subunit. They have loop connecting them to each other. The loops are connecting them. Loops like this are connecting them to each other. And you can see that they are 4 colored. They are the 4 chains. We have this color is different from this color, different from this color, and it is different from this color. You notice this color too. We have 4 different colors that are indicating the 4 chains. Rotating this, click it and rotate. This disc shape that you're looking at is nested in each chain, and they are the hemi group. Each of the hemi sits in a pocket formed by the protein, and it is cradled by the hydrophobic residue that holds it in the right orientation for oxygen binding. This is to tell you that the shape and the function are inseparable. Now that we have explored this report, let's go back to the main page. And over here, you can click to download your file. Click. Ensure that you download it in the PDB format. And you have just successfully downloaded your first PDB structure file. Now we can open the downloaded PDB structure file. It's a plain text file. You have to study it and check through all the columns to understand it, understand the structure you are downloading, check for the remarks, and every other important details in the structure. So this is us exploring it. At the top, you will see the header. It contains the name of the molecule, 4-HHB, and the date it was deposited, 7th of March, 84. The remark contains, let's look for the remark. These are the title, the caveat, read through it. The remark, it contains notes from the author about the data quality, about the experimental condition, and any unusual feature. All of these things are worth reading when we are doing a serious analysis. Now you should also scroll down by looking for where the atoms are. Atom, column, scroll down till you see atom. Okay, over here we have atom. Over here we have atom. Here is the atom column. Atom, 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 atom. Yeah, let me stop right here where I can see B and I can see A too. So I'm looking for a one where I see A and B to explain what I need to explain. So in this atom column, each of these atom line describe one atom in the protein. We have the atom serial number. We have the residue name. We have the this is atom serial number. This is the atom name. This is the residue name. This is the chain ID A. This is the residue sequence, the sequence number of residue. Then we have these three different columns that contain numbers. They are coordinates in action. These coordinates are literally where the atom sits in the 3D space. And this other column that we have 1.0 is called occupancy column. It's usually 1.0, which tells us that the atom was found in this position in all unit cell of the crystal. Then this last column is the big factor or temperature factor, the B factor or temperature factor. It's a measure of how much the atom vibrates or is disordered. And a high B factor means that the region of that protein is flexible. Let's also find the HET ATM that we mentioned earlier. It has the same format, but let's look for it to explore what it looks like. Scroll through, scroll through, scroll through, scroll through, scroll through. We have a lot of atoms, so you have to be patient. We have the HETA column. So over here, you see the same thing. It has the same format, but these are for heteroatoms. They are non-standard residue. So check out HEM. This HEM in the residue name column. These are the Hemi atoms. You will notice that they are in their own chain entry too. This one is in MED chain. This one is Hemi chain C. This one is Hemi chain B. They are in their chain like that, positioned where you saw them in the D structure you saw in the viewer earlier. And with all of this in check, we have successfully learned how to retrieve a 3D structure and how to read the raw PD file, understanding every column. I'll walk you through the steps again. Open your browser, go to rcsb.org. We're going to be searching for human hemoglobin like we did. We search and we saw it. Explore the page, check out for the macromolecule section, check out for the chains, check out for the ligand section, check out for the hemi group, understand the concept of hemi group. This is the ligand section. Each hemi group per chain. Go back to the report, explore the image that comes up when you check the reports, the 3D reports. And after that is checked, you download your file over here. When you download your file, read the file just as it is in the report. Find the atom, find the HET, ATM line. Check the title to be sure that it is correct, and every other details that you need to check. At a more advanced level, we can use AlphaFold when there is no experimental structure. It predicts the 3D structure from the amino acid sequence, like we said earlier. And at a more advanced level, we pull this data programmatically, retrieving with BioPython. We load this structure into PyMol, and that's where the reanalysis begins. We measure the distance between atoms, we identify mutation sites, we compare two structures by aligning them, and we ask whether a disease-causing mutation actually changes the shape of the protein. This retrieval step that I've gone through is basically the prerequisite for all of that. So go back over the video, take pauses when you need to, practice along, understand the concept you're working with, and I will see you in the next video.